Sí, mucha mierda.
Mucha mierda, mierda, mierda.
Good morning to everyone. Good morning. My name is Francisco. I am from Ecuador. I am an environmental engineer, and I am taking the ECODIM course at Realizado. I have the pleasure to introduce you a well-known scientist called Dr. Pedrós-Alió. He is a microbiome ecologist. He got a PhD in bacteriology at the University of Wisconsin-Madison. He is a research professor at the Marine Science Institute, Consejo Superior de Investigación in Barcelona, Spain. His current work involves genomics of marine bacteria, diversity of bacterial communities, extreme environment, salares in Atacama, northern Chile, thermal springs from Costa Rica to Antarctica. His presentation prepared for this morning is about dipping into the biosphere. Thank you, Carlos, for coming to this symposium and for sharing your experience with us. Welcome to Concepción.
Thank you. I'm very glad to be back here. I was here, I don't know how many years ago, at this very place giving another lecture, and it's nice to be back again. But especially for two reasons. One is that I have been following the ECODIM course for many years, and finally I've got to be here personally. So I thank Kurt and Osvaldo and Rodrigo and Nicole, and I don't know whether anybody else was involved, for inviting me to this meeting. And the second thing is that it's in the symposium today is in honor of Carl Woese. And Carl Woese, of course, has been very important for biology. He's created one of the most important revolutions in biology. And I was a PhD student in Madison when in the late 70s, when he came up with this idea about archaea. And he came to Madison to give a talk about this. And so I'll be telling you a story about this tomorrow, to those of you who come to the Chato, because he not only caused a revolution in science, but he also caused a religious conversion. But you'll have to come to the Chato to listen to that story. Okay, so this is our task, this ocean full of microbes. I almost said prokaryotes, but no. Full of microbes. And we would like to know how many are there, how many different species there are, what different functions do they have, what can they do, and how does this work. And so I'll divide the talk into 8 sections, but one I skipped, so it'll be 7. And the first one is to convince you that there are a lot of bacteria in the oceans. Actually, Linda already mentioned the number, so you'll see the number again. But I think this paper in 1959 is the beginning of modern marine microbiology, because in this paper, Janesk and Jones counted in a vertical profile in the ocean, down to 160 meters, the number of bacteria with different methods. Plate counts and some direct counts and microcolonies and some direct counts. And as you can see, the number of bacteria is here in a log scale. And the difference in numbers was amazing, just several orders of magnitude depending on the method you use to count them. But in the 70s, late 70s, the epifluorescence technique came by, and then it turned out that the number was 10 to the 5th bacteria per milliliter. So this was the beginning of people realizing that microbes in the ocean were important. And the thing is that to come up to this number that Linda showed, I need something more intuitive. And so we'll be moving from 1 cubic centimeter of seawater up to the whole ocean. And you consider 1 cubic centimeter, you have these 10 to the 7, prokaryotic cells. And if you look under the microscope, this is what you see. You see little croissants and croquettes and meatballs and so on, but you don't know how many species they belong to. You just can count them, and that's it. Then if you move to 1 cubic decimeter, you have 10 to the 8. And this is many times the volume of water that is used to build clone libraries. Now, 10 to the 8 individuals are a lot of individuals. And if you were studying forests and trees, you would have to consider that maybe you're mixing different ecosystems. Of course, the forests of the Patagonian Andes are dominated by one species of tree, and the Valdivian forest has one of the largest diversities of trees in the world. And maybe we're mixing these different types of ecosystems. So when thinking about how microbes live, we have to keep in mind these kind of problems. But then if we move to a 1 cubic meter, then we have 10 to the 11 cells. This is approximately the volume that Craig Venter used for his pilot study in the Sargasso Sea. And if we were botanists and we wanted to study a forest with this number of individual trees, we would have to analyze a forest the size of Mexico. So again, you have to think in that what we're dealing with is very different from the things we're used to deal with large animals and plants. And then let's move to the whole ocean. And we come up to this number, 10 to the 29th. And that's such a large number that just like Linda did, I like to spell it out with all the numbers of zeros. Now you have to remember that we have 10 to the 11th neurons in our brain, and that there are 10 to the 11th stars in our galaxy. And 11 is somewhere in here, right? And you still have all these numbers to go up to the number of microbial cells in the oceans. So I like to say that this is not an astronomical number, but a meta-astronomical number, because it goes far beyond the numbers of stars. So imagine how many species can there be, represented by maybe 1 billion cells or 1 million cells. The number can be really very large. And of course, it would be nice to know. Many people say, okay, bacteria, there are many, but they're so small that they don't count very much. So let's compare them to the favorite marine animal of most people, the whale. Here you have a whale. And if you compare whales and bacteria, of course, length, the whale is pretty big. The bacteria are pretty small. If you look at the mass, the difference is even larger. But then if you look at the numbers, and here for whales, I've counted minke whales, which are the smallest and the most abundant ones. And for the mass, I have counted the blue whales, which are the largest ones. So I'm magnifying the biomass of the whales. And this is the number of microbes that you saw before. So this is the biomass of whales, the biomass of bacteria, and then by a simple mathematical division, you come up with this. The biomass of bacteria is equivalent to 56,000 million blue whales, or the biomass is 10,000 times the biomass of whales. So it's not only that there are many, but in terms of biomass, whales are irrelevant. I'm sorry. Okay, the next question is, who is with the carbon cycle? Because then you can say, okay, there are many and there's a lot of biomass, but they don't do anything interesting. So this is the second beginning of marine microbial ecology. The first one was Jana's paper, and this one by Pomeroy in '74 was one of the first papers demonstrating that the action was in the smallest size fractions. This was the marine food web as known at the time. There was the phytoplankton that was collected in nets, and it was eaten by zooplankton, eaten by fish and top carnivores such as ourselves. And there were also some smaller phytoplankton, this nanoplankton, that was eaten by other organisms, but that was it. Now Pomeroy fractionated the water samples and measured respiration in these different size fractions and realized that there was a whole world outside of this food web with organic matter excreted by all these other organisms that was used by bacteria, eaten by protists, and back into the web. And he could show that not only bacteria were very abundant, but they were responsible for most of the action. In fact, if we organize marine food webs by size, from the smallest to the biggest, this is a fish larva, it's not even an adult fish, so the whale would be 2 or 3 floors up. And we have here the heterotrophic organisms, and here we have the autotrophic organisms, the phytoplankton. Then we can do some estimates and see that these phototrophic microorganisms are responsible for 50% of the primary production on the planet. That means that half of the oxygen molecules that you all are respiring at this very moment come from marine microorganisms, and the other half from land plants. So they are not a minor thing. And if you look at respiration, then you consider all the organisms, and it turns out that more than 95% of the respiration in the oceans is due to the microorganisms. So in terms of carbon fluxes, in terms of climate regulation and everything, whales, again, are irrelevant. The important stuff in the small size fractions. OK, so we know there are many, and we know they are very important in terms of the carbon cycle. But we would also like to know how many species are there. Now, I have many colleagues who say, I don't care who's out there. I just care about what they do. They're fixed on the function of the organisms. I mean, that's fine. But I do want to know who is out there. I think it's very different. If we could have one autotroph and one heterotroph, and we would have a functional carbon cycle. Why do we have many species of each? And I think this is an interesting question from many different points of view. So I would like to know how many species are there. Remember that this is the number of cells, a huge number. And now I'll show you 3 different estimates of the number of bacterial species in the oceans. This one, by Hagstrom and colleagues, 2002, they said a few thousand. This was based on conventional clone libraries. This other one, Curtis et al., the same year, they said 1 million. And this was based on some theoretical assumption, and then a calculation with some numbers. So it was a completely different approach. And finally, this was another one a bit earlier by Dick Wison, and he came out with 1 million species. So when you have an estimate of something that runs all these orders of magnitude, it means that we don't have the slightest idea. So can we do something about this? OK, the problem is this one. If we look at organisms that have a size similar to our size, like, for example, 2 seagulls, and these are actually, no, I think they're Argentinian. Sorry. But you also have these 2 species here in Chile, so I guess I'm fine. You can look at them and you immediately see that they are different, but you also see that they are related. It's relatively easy to find these kind of things with organisms that are our size. But in bacteria, as we were looking at before, this is not possible. So we don't know whether all of these cells belong to the one species or whether each one is a different species. And here comes the contribution of Carl Woese, because, of course, here you have him painting these catalogs that Norm was telling us about before, and separating the ribosomal RNA. And this idea, and not only the idea, but the methodology to come up with this, as I was saying before, it was a revolution in biology. It completely changed the paradigm, and all of a sudden things that were not possible to fit together, all of a sudden fit together. But the second step, Norm was very modest, but he was responsible for taking all this approach and all this philosophy into the field. And thanks to his contribution and his lab, we are able to use these techniques in the field. So that was the second thing. In fact, I was at a meeting in Ljubljana in the middle 70s, and Norm gave a talk, and I had my second religious experience and said, this is the future of microbial ecology. So then a few years later, I went to his lab, and it was at the time that the science paper with the phylogenetic stains came out. And I noticed that there were a lot of science and nature papers coming out of this lab. So at one point, I asked Norm, what is going on here? You're only publishing in Nature and Science. And for a minute, he thought maybe what have I been doing wrong? So eventually he said, but there is nothing intrinsically wrong with it, is there? Certainly, there is nothing wrong with it. OK, so then the next, the criticism that comes from botanists and zoologists is they say, OK, you guys using molecular methods, you're overestimating diversity. You find a different sequence and you call it a different species, but you don't really know whether it's a species or not. And in many cases, that's true. We don't know. But there is one beautiful example where we can compare the molecular methods with the conventional definition of a species, the biological species concept. And the example I will show you was done in Naples by Amato and other colleagues. They've been studying the Bay of Naples for many decades. They have a lot of clones that have been isolated from diatoms. They have a lot of annual cycles and everything, so they know the system extremely well. And morphologically, they could tell there were 2 different species of this diatom called Pseudonychia. So they could, I'm sure you cannot distinguish it. I cannot either. But they had an expert eye, and they could tell them apart. So by light microscopy, using the morphological species concept, there were 2 species of Pseudonychia. So they said, OK, let's look at the ribosomal RNA. And they looked at the large subunit ribosomal RNA, and they, with this phylogenetic species concept, and they could see that there were 1, 2, 3, 4 within this morphotype, and 1, 2, 3 in this morphotype. So at this point, the botanists and the zoologists will say, see, you are overestimating diversity. However, they did something more. They also looked at the interspecies regions between the ribosomal RNA genes, which, as you know, are a lot more variable than the ribosomal genes. And they found that in this case, it was the same, 3 again, but in this case, they had a further division here of 2. So again, overestimating diversity. Well, of course, the beauty of this example is that diatoms do have sex. Therefore, you can test the biological species concept by crossing them and see when they have a fertile descendant. And they did that with a painstaking job of mixing many different clones and looking at the difference. And what they saw with the biological species concept is that it fit very nicely the interspecies region. So even the ribosomal RNA was underestimating diversity, not the other way around. And then they looked very carefully at the morphology with the electron microscope, and they could see tiny differences in the fruit stools that fit with these different genotypes. So they could define many more different species. So the lesson to take home from this is that if we are using ribosomal RNA, we are not overestimating diversity. We are on the safe side. If anything, we are underestimating diversity. Okay. Now, if you organize the individuals and species in a community in a rank abundance curve, and you put this most abundant species here with this number of individuals, and then the second one, the third one, this is the kind of curve that you get. And Linda showed this before. And as she said, Mitsuki named this area here the rare biosphere, which is a very nice sounding name. So it's nice to use and so on. And the techniques we have available to look at the diversity along this curve are molecular techniques and pure culture techniques. If we use molecular techniques, we usually look at the most abundant organisms. And this is because you're always using primers. And of course, the primers, if one organism is 10 times more abundant than another, will be busy looking at this abundant organism. And only occasionally will find a sequence for the rare organism. So that's why molecular methods usually look at the most abundant ones. And the pure culture techniques bring up some of the organisms that like to grow in the culture media that we use. But they may be very rare, or maybe middle, or maybe more abundant. The problem is that there are only a few of them that we are able to grow in pure culture. So as molecular methods have improved, we've been able to push along this curve towards the rare biosphere. You use a fingerprinting technique. You see maybe around 30 taxa or so on. If you use cloning, you see a few more. If you use metagenomics, you see quite a few more. And if you use the method that Linda explained of massive parallel sequencing after a PCR of the 16S, then you get many more taxa. But how far along this curve are we? This is something that I would like to know, so that we can bracket what is the diversity of bacteria in the oceans. So we would like to know what's the length of this curve. If you use a conventional cloning method, was used for many years, what you see is this thing here. You have here the number of clones, and maybe you guys in the back don't see the numbers, but this is 300. So after examining 300 clones in different communities, you get up to 80 different phylotypes, or 40 this order of magnitude. And if you use estimators to estimate how many different taxa would there be, the numbers are around 300, 100, 300. So this is what you get. Now, if you use the modern methods of massive parallel sequencing, what's the image that you get? And the samples I'm going to show you are from this area of the world. This is Blanes. Blanes is about 40 kilometers north of Barcelona in the Mediterranean, and France is around here. And we have a regular station that we sample every month here, this station here. And then this is a cruise that we did, transect and vertical profiles. And I'm going to be showing you data from this sample here, 5 meters open ocean, and this one here, 2,000 meters of open ocean. So this is what it looks like for the same type of core for these methods of pyrosequencing. Here you have 15,000, 20,000. So it's an order of 2 orders of magnitude more. This 200, 20,000. 2 orders of magnitude more. And the number of phylotypes that you get is around 1,000, so it's about 1 order of magnitude more. So we get more sequences, and we get many more taxa. But remember that the estimates here were 300. So obviously, they were completely wrong. So one has to be very careful with his estimates, because many times they depend more on the clone library that you have built rather than on nature or the real diversity of nature. OK, well, so what we did was to do many sequences of these 2 samples, up to 500,000. This means a whole pyrosequencing plate for this sample and another whole pyrosequencing plate for this sample, so it was very expensive. But I really wanted to do this. I wanted to see what happened. And what you see is that the numbers, the curves, begin to stabilize. And this is something around maybe not, well, 2,000 or something, and here maybe about 4,000. So the bottom sample is more rich in species than the surface one. But it looks like with these numbers of sequences, we're getting somewhere. We did a fit, this Weibull function fit, and you don't see probably the difference, but there's a black line with the experimental data, so the fit is very nice. And it's so nice that we dared to estimate what would be the asymptote and how many more sequences we need to get to this number of phylogenetic phylotypes. To get 99% of the phylotypes, we would need 2 million additional sequences. And to get 99.9, we will need 5 million sequences. So this is not unthinkable. But it means a lot of effort for just a little bit of additional species. So the thing is, if this looks like we're getting to the end of the rare biosphere, let's compare it to the cultures to see whether we find all the cultures. If we're really getting to the end of this curve, we should see all the cultures that we have isolated from the sample. We should see them in the sequences. So let's see whether that's the case. We isolated, I think it was 400 different cultures from the same from the surface sample, and we compare them to the sequences. And what you have here is the number of the rank of the cultures in our culture collection, the number of times that they appeared in the sequences, in the pyrosequencing sequences. And this was the organism, the culture that was found more times in the sequences, Erythrobacter, and it was found both at the surface and at the bottom. These 2 ones were not found in any of the 2 sequence collections. So there was cultures isolated from the surface sample, and we couldn't find them after 500,000 sequences from the surface or 500 sequences from the bottom. Here's another one that was found in both places. These ones were not found. These ones were found at the bottom, even though they were isolated from the surface. So as you can see, there are many cultures, all of these in white dots, that we could not find with all these many sequences. So as you get more and more sequences, you get more of the cultures in your sequences. But as you can see, again, we have this kind of curve, and also this will go up. Look at this. This is only 24% of the cultures were found. Only 36% of the cultures were found. So most of the cultures could not be found. In other words, if this is the surface sample, and this is the bottom sample, this is the rank abundance curve, this is the number of sequences we had, 500,000. This is what we would need to get 99% of the sequences or the phylotypes, and this is what we need to get 99.9% of all the phylotypes. And here we got only 24% of the cultures, and 76% of the cultures are in this rare biosphere. So we still have a long way to go. Now think that all of these organisms have their genomes, and their genes can do different things, and we don't know what they do. So potentially, there are a lot of functions that are hidden in all this rare biosphere. Now, let's look at a few properties of these bacteria. If you look at this curve again, you assume that the abundant ones are abundant because they are growing actively, and they are contributing to the carbon flow and that kind of thing. And if they are growing actively, they're experiencing predation and viral-lysis. So this is the dynamic portion of the ecosystem. This is running the ecosystem. On the other hand, the bacteria here may be dying and maybe become extinct. And also, there is dispersal. There will be migration. They will be coming here. But they're probably not growing very much. And in the case of the most abundant ones, you have to think whether they are resistant or not. And I'll show you an example of an experiment that indicates that maybe this is the case. And I think one of the groups is doing a very similar experiment to this one. This was done by Bouvier and Del Giorgio. And what they did was collecting water from the ocean and incubating them here. And they could see with fish the different types of bacteria, alpha, beta, gamma, Bacteroidetes, and so on. Then when they incubated, this was the initial sample. When they incubated the sample with viruses, this is what they got. Gamma proteobacteria increased a lot, Bacteroidetes disappeared, and beta bacteria, proteobacteria appeared a little bit. But gammas were winning the competition. However, when they did the same experiment resuspending the bacteria in water without viruses, what happened was that the beta proteobacteria were growing and the gammas were not there. So this is suggesting that beta proteobacteria were active in nature, but viruses were getting them real hard. And why this is the case, I don't know. But it just suggests that we have to be very careful in thinking what is important in nature, whether predation, viral lysis, or growth is more important in determining the composition of the community. The second thing is that you would think that these bacteria that are abundant have a significant contribution to carbon flow. The intermediate ones could have a significant contribution to some particular element flows, for example, nitrogen fixation. You don't need to be very abundant to cause a significant impact in an ecosystem if you fix nitrogen. So you can have an intermediate abundance, be important in this kind of process. And these ones here probably don't have any contribution to carbon flow because they're just too rare. But they contribute to biodiversity, and potentially they can contribute to the carbon and element flows if conditions changed. And the nice thing about this is that if you're a bacterium that is declining because conditions are changing and you don't like them, you cross several thresholds. One of these thresholds is the fact that viruses need a minimal concentration to be successful in attacking a prey. The problem is that they move by Brownian motion, and they have to find their susceptible cells at random. If the concentration of susceptible cells is below a certain threshold, they never find them, and therefore there is no viral lysis. So by being rare, you are protected from viral lysis, in principle. And this is one number that came out with some E. coli viruses, but there must be a threshold for the other bacteria also. And another threshold you cross is the one of predation, because predators are not silly, and they like to eat the bigger cells, which are more nutritious and more active. They don't like the small cells, which are not very nutritious because they are not growing. So even though, in principle, they are not selective, in many cases has been shown that they do prefer to collect the biggest cells. So therefore, if you're rare, you are safe from predation and from viral lysis. So you may not be growing very fast, but you're not dying either. And you can stay here for a very, very, very long time, which is a nice property of this rare biosphere. Now I'm going to tell you the sad and short history of the Pouli. These are the Hawaiian Islands. And as you see, there is a green part of the islands where the winds come, and a dry part of the islands. And these forests in the green part are actually, it's amazing that there's not much known about them. But they, of course, are threatened by all the invasive species and this kind of stuff. So there were some students from the University of Hawaii that decided to study the biodiversity, actually let me go back, of this forest here in the island of Maui. And the birds in these islands, there are not very many different birds, but there is one particular group that has experienced this incredible diversification. And this is the finches of the Hawaiian finches. So look at these different species of finches. Aside from the colors, look at the bills. There are bills which are very different. This one is incredible. This one uses the lower portion is tough, and so it hits the wood like a woodpecker. And then the upper portion is flexible and long, and he puts it in the hole and grabs the worms and eats them. So it's like a Swiss Army knife of a bill. So I think if Darwin had visited Hawaii instead of the Galapagos, nobody would be talking about Galapagos finches. They're very boring. They're all brownish and so on, and they're very similar. Look at these guys. These are really pretty. Okay, so the Pouli is this one here. And what happened was that at this time in the 1960s, the students were studying the diversity of this forest, and they saw this bird, and they realized it was a new species. This is very strange. To discover a new species of a bird in the 20th century is really exceptional. And to discover it in Hawaii, that has been so well studied, was even more exceptional. So they were really excited. And the first thing they did was to shoot 3 of them, because you need to have type specimens. And to do that, you need to kill some of the birds and stuff them and put them in the collections. They, of course, were not aware of how a large proportion of the total population they were killing by killing 3 individuals at the time. Otherwise, I'm sure they would not have done it. But at any rate, I think this is the only picture of a pouli in the wild that there is available. So they kept studying this bird. You have here the number of individuals per square kilometer. This is 1975. As you can see, the estimates were going down, and they got to a point where the estimate was 3. So they only knew about 3 individuals of this species in the wild. And this is really worrying. It looks like this species doesn't have much to do. This is obviously a very rare species. It will belong to the rare biosphere, no doubt about it. So there was another problem. It was that every individual was in a different territory. So these are the altitude in Maui, in this area of Maui. And one of the individuals was here. Another one was here, another one was here. So there was no way they could mate and reproduce. So they decided we have to grab one of these individuals and bring it to the territory of the other one and hope they like each other and that things function properly and so on. So they did some studies of the sex by collecting a feather and sexing the individuals, and they found that this was a male and these 2 were females. So they said, okay, we'll collect this female and we'll bring it to this male's territory, because in theory a male likes to stay in his territory and a female doesn't mind moving to another place if the male is attractive enough. Okay, so they did a lot of studies with other species, trying to see the best method to collect this bird without harming it and bringing it to the other place and that kind of thing. Eventually, everything was ready, and at one night, they collected this female, and through this tract, you have to imagine that this is a very wet forest. It's very steep. It's raining all the time. It's extremely cold. It's at night, and you're struggling there, bringing this little bird, trying to protect it and everything. So they put it here. And they instrumented it so they could follow the bird. So next morning, they were all expectant, seeing what would happen. Would they like each other? And what happened was that this bird just flew back to its own territory. Okay, so what do we do now? So what they decided to do is we have to collect the 3 birds and try to reproduce them in captivity. And they tried to do that, but they only could collect this one. The other 2 disappeared. Nothing else was known about them. So this poor guy was the only single known individual of this one species. And the scientists looked at the individual, and they saw it was very old, so it was unlikely they could reproduce anymore. It was sick. There was an eye missing. And worse, it was another male. So no wonder it had gone back to its territory and hadn't stayed there. Of course, sexing by feathers is not a very precise technique, and that was the problem. When they did it with the blood, then it was realized that there were 2 males. Unfortunately, this pouli died, and the species became extinct. The reproduction in captivity has functioned for other Hawaiian birds like the akepa. Look at this little thing. That's the chick, and this slightly larger chick. They're very cute, and it's nice to have all this diversity. I mean, it's so sad to see a species become extinct in your hands. And this is what happened to the pouli. Now, the reason I have told this story, let's go back to bacteria, is that this will never happen to a bacterium. Because that one single individual can reproduce, and if things are okay, could actually colonize the whole planet. So when you think about the rare biosphere, you have to realize that the ecology of these rare bacteria is completely different from the ecology of rare species in animals and plants. And that the possibility to be there for a long time, doing nothing, waiting for a second opportunity, is so large that maybe this tail is longer and long and long and long and long. We don't know. Okay, so we have these 2 thresholds. But if you decide to grow because conditions change and you move along this direction, you again cross these thresholds, and then you become susceptible to viruses and predation. But at least you're growing. I mean, you have to grow sometime. And I'm going to show you an example of this. This was a study done in the Rías of Galicia in northwestern Spain, when we had this oil tanker, the Prestige, that sank, and the contamination was awful. And then the Spanish authorities realized they needed to do some research on oil degradation. They hadn't thought about it before, of course, but then there was a lot of money to work on this. So everybody, all of us were experts in oil degradation for a while. At any rate, this experiment was done there, and what people did was to use oil derivatives and add them to different mesocosms and see how things would evolve. And what I want to show to you is this one example of one bacterium, this Psychoclasticus. There was a fish probe developed for this bacterium, and with time, this is days, in a control mesocosms without oil, it's the black dots, so essentially zero, not detectable. And in the experimental mesocosms with some oil derivatives, it goes up and then goes down. And you see the oil derivatives, they go down, and when they disappear, then this guy goes down, right? So it seems like it's a very tight relationship. Now, if you have more amount of oil derivatives, you have a larger peak, and a larger one, and a larger one. So it seems clear that this guy was sitting there in the red biosphere, conditions change, become adequate for it, so it can grow, it can cause a significant impact in the carbon cycle, and once this substrate is gone, it goes back into the red biosphere, probably because viruses and predators are getting it. So this is probably happening all the time in nature. So this means that the situation of this rank abundance curve is very dynamic. In fact, you can look at this by obtaining this curve, not by looking at the DNA, but by looking directly at the ribosomal RNA. As you know, the amount of ribosomes in a cell is generally proportional to the growth rate, because if you grow very fast, you need a lot of ribosomes to synthesize enough proteins. If you grow very slow, you need so many. So in theory, you have one copy of the DNA per cell, but you can have many copies of the RNA if you're very active. So if you compare, in this case, the abundance of the different taxa from DNA with that from RNA, you get more or less a straight line. But there are some points which are above the line. These have more RNA than they should have compared to the DNA, and these ones have less. So these ones are less active, and these ones are more active. So in this case, this is the DNA curve. If this organism is more abundant in the RNA than in the DNA, it means it's moving this way. If this one is less abundant, it means it's not very active and is moving this way. So this curve is very dynamic and active all the time. And bacteria are moving back and forth between the rare biosphere and the abundant cells. So why would want to be rare? And one reason is that bacteria that decay from blooms from the past are here when conditions are not adequate. This is the example I showed you, but I'll show you another example. So at some point, conditions are adequate for you, you grow, you become abundant, but then conditions change, you cannot grow anymore, and you go back into the rare biosphere. Another one is the dispersal by specialists that I will talk about later, put an example. And another one is dispersal and immigration from other sites. So I'll put one example of each. The first one is from the Arctic. This is the Amundsen, a Canadian icebreaker, went up to the Arctic in the fall and allowed the ocean to freeze around it, so we could follow the community in the Arctic winter for a whole year, which was something fantastic, something unique. And one thing that Laura Alonso found, I was talking about Laura Alonso with a couple of you guys before. She's my first academic granddaughter, and I guess grandparents have a weakness for the first grandchild, so I have a weakness for her, but she deserves it. So at any rate, Laura was looking at bacteria, and at one particular point, she noticed that many of the cells were very large, and they were forming these aggregates. So what she did was to get a clone library from that sample, from that time, and in effect, many of the clones, most of the clones, belong to this beta proteobacterium, Xanthinobacterium. So there was a case of a natural bloom. The case before, it was an experiment. We were manipulating things and that kind of thing, but in this case, it was a natural bloom. So she developed a fish probe for this one bacterium, and along the time, this is day of the year, so this is January 10th, and this is February. You can see that the bacterium is not there, goes up, and then goes down again. That was in the middle of the winter. It was dark, it was cold, and everything. And there was this organism, for some reason, found conditions adequate, grew, became abundant, important in carbon cycle, and then went back. So as I was saying before, this is probably happening all the time. The second case is specialists that are swimming around. And a very good example is the Vibrio fischeri that grows in the light organs of this squid, this bobtailed squid. In the light organ, there are about 10 to the 10 cells in 1 milliliter. So there are a lot of them. And in the waters, they are maybe between 10 and 10 to the third per milliliter. If you think that there are about 10 to the 6th or 10 to the 5th cells per milliliter in total, this means these guys are rare. They're part of this rare biosphere. And what these squids do is that at night, they move from the sediment up and they feed. And these light organs, apparently, they're in the lower portion, so that if a predator is looking up against the sky, which is lighter, maybe with the moon or with the stars, this light compensates and it doesn't see the shade. So it's used to defend themselves from predators. But in the morning, they go back down to the sediment and they stay there for the whole day. Of course, the bacteria are fed during the, when they go down in the sediments, they feed the bacteria so they can grow for the next night, so that they have abundant bacteria. But if this were happening all the time, the organ would be enormous, because bacteria like to grow. So if you feed them, they keep growing forever. So every morning, before going back to the sediments, they excrete most of the bacteria. They excrete a lot of the bacteria in the light organ. And then during the night, they're growing slowly back up. So as you can see, these bacteria here are coming from these organs. Every morning they are thrown away, and then they form part of the rare biosphere. In a study in Hawaii, the scientists could see that in the 2 bays where the squid was present, these bacteria were found in larger concentrations than in the bays where it wasn't present, where the squid was not present. So obviously, the source of these bacteria are this squid. The same thing happened in Australia in a different study. This bay had the squid, the others didn't, or not as much. And also here in the morning, the concentration is high. At night, it's lower, because they have been dispersed. In the morning, the squids throw away the bacteria, the concentration is high, and then slowly they are dispersed and the concentration goes down. So here we have a very clear example of a bacterium that forms part of the rare biosphere, because the water is not its place for growth. Its place for growth is a very specific one in one animal. Now, the thing is that if these bacteria were not in the water column, this system would not work, because the baby squids are not born with the bacteria. They have to collect them from nature. And the way this happens is that they have a very specific sensor that detects when a Vibrio fischeri comes by. And when the Vibrio fischeri comes by, collects it and forms the light organ and grows the population. And this is extremely specific. Without Vibrio fischeri, it doesn't work. It only works with Vibrio fischeri. So you need this population of rare bacteria to keep the symbiosis going. And the last example is the one of dispersal from other places. Here we have, this is Africa and the Canary Islands. And you see this dust storm taking a lot of dust from the desert into the oceans. And another one of my academic descendants did this study. It's a beautiful study. What he did was to collect dust in Mauritania and then go to the Pyrenees between Spain and France in a high mountain lake and put a system to collect deposition. So this is a bucket, and it collects the dry deposition. When it rains, there is a sensor, and this lid is put over here, and then it collects wet deposition. So you can see what comes with dust and what comes with rain. Then he also looked at the organisms in the surface of the lake and within the lake. And what he found was that some bacteria, in this case, were identical in Mauritania and in the Pyrenees. So you can see these are basically identical. So the same bacterium was living in the soil in the Sahara, and it had been transported to this lake in the Pyrenees. The same bacterium was dispersed from one place to the other. Of course, if this bacterium doesn't find the appropriate conditions in the ecosystem, it will not grow. It will form part of the rare biosphere. And that's what he found in the water of the lake. It was there, but in very low abundances. The nice thing is that he did some experiments where he collected this dust and inoculated water from the lake. And this bacteria could not grow. But if he killed the autochthonous bacteria, if he killed the bacteria from the lake and then added this dust, then these bacteria were able to grow. So again, this is a beautiful example of dispersal from one place to the other. The organism could grow there, but it could not compete with the local microbiota, and therefore form part of the rare biosphere. However, if conditions change, there's always the potential that it can grow. So the last question is this issue of dispersal. How far can it go? We thought that the best way to study this was to use the two polar zones, because they have similar conditions, but they are very far away, and they're separated by warm areas, so that probably organisms adapted to live here or here cannot survive in this place. So we thought it was a nice system. And there are different ways that this dispersal can proceed. For example, deep ocean currents move from one side to the other. This takes hundreds or thousands of years, so it's very slow. Another possibility is through air. Since bacteria tend to float, maybe they can go from one place to the other. And a third one is with birds. For example, the Arctic tern breathes here, but then it moves down to Antarctica in the northern winter. So there are different mechanisms where bacteria can move from one side to the other. And we wanted to know how similar were the bacterial communities in the two polar zones. So we used the ICOM dataset. Again, another example of how useful it is, because we only collected a few of the samples, but most of the samples were collected by other people. And we had these pink samples in Antarctica, these blue ones in the Arctic, and the gray ones in temperate zones. I'm going to show just two slides. One is showing how similar were the communities. And as you can see, only 40% of the phylotypes were common to both poles. So most of them were different in one pole and in the other. However, when we compare communities, this is a complex graph, and I will only explain one part of it. Look at this. These are surface waters from temperate areas, the Atlantic, the Mediterranean, the South Pacific, and so on. And these here in color are the polar samples. So the polar samples fit together in one cluster, which is completely different from the cluster of temperate waters. So even though they only share 14% of the OTUs, they are more similar among themselves than with the temperate waters. And the other thing is that if you look at, for example, coastal waters in summer, you have here from the Southern Ocean in red, from the Arctic Ocean in blue. They're closer together than any others, but they are different. The same thing for coastal waters in winter, and so on. So there's a hierarchy of similarities here, where communities are more similar among themselves, but they're different from the rest. So dispersal is possible, but there's still a lot of things that make differences between the communities in one side and the other. And the last topic I want to talk about is this aquae calidae, which I like to call our studies in warm environments. We do this, well, these are places where we take samples, but the ones for aquae calidae are mostly here, with colleagues in Costa Rica, Lorena Uribe, and colleagues in northern Chile, Cecilia de Mergasso and Lorena Escudero, in central and southern Chile, Beatriz Díez and Mónica Vázquez, and these samples were collected by Bea and Mónica in Antarctica. And all along this area, we have thermal springs, and so we think that the communities in these thermal springs must be similar, and they're like hot islands in the middle of a cold ocean. And so we can study questions of dispersal. Besides that, this takes you to very nice places. For example, here in El Tatio, these wonderful microbial mats, and also to some dangerous sampling expeditions. This is in Huinay, in southern Chile. This is a fjord, and this is the station San Ignacio del Huinay. We sampled here one station, one thermal spring, here another one, another one. I'm going to show you a few slides of this one, just to have a little bit of tourism to finish the talk. This is the Google image of the area. This is the fjord, and you have to go up this river here up to the geyser. And for this, you have to cross the river 5 times, and climb up a hill with a rope, and then climb back the other side. So it's quite something. And some of the crossing of the rivers are not trivial, especially if you're carrying all the samples on your back. You really feel it's dangerous. So you will have to forgive me for showing the next slide, which is just a tourist slide. But I was so happy that I had been able to cross the river 5 times and getting there to get the samples that I just had to show this slide to you. At any rate, the communities here are very interesting. And for example, one of the things we look at is the microspatial diversity. This is a microbial mat. Here's the hand of the researcher. And we collected samples on all of these places. Look at the temperatures. Here is 57, 55, 52. And here is 59, 60. So the difference is not that much. It's like 5 degrees or so. But you see the color changes, which is already an indication that the community changes. And after sequencing by Illumina, this is the same structure of samples. And you can see how cyanobacteria in green dominate at the lower temperatures. And Chloroflexaceae in orange dominate at the higher temperatures. So this micro-distribution is one of the things we like to look at. But then we also look at the differences between the different springs. Those 3 springs I showed you in Winai. These 2 are close together, and they are similar. This one is further away and is less similar. So again, here we look at the similarities across a spatial scale of a few kilometers. And then, if we look at total samples from Costa Rica to Antarctica, this is what we see. And I realize this is not telling you anything, but when we put the temperatures, then it becomes more interesting. So the high temperatures, they're very similar regardless of where they come from. One of these is from Costa Rica, another one is from El Tatio, and another one is from Winai. So regardless of the area where they are, the hot ones are very similar. So this means dispersal is not a problem at all. They're able to move from one side to the other. However, when you look at lower temperatures, 50s here, 40s here, and so on, you see 2 different patterns. And these belong to the north, to Costa Rica, and these belong to the south, to Chile, and Antarctica is here. So this is something interesting to me. I don't know what it means. The data are very recent, but if anybody has some interpretation, I would like to listen about it. So the final considerations are, one is publicity of this book. I published this year, La Vida al Límite, and it's about life in extreme environments. It's a very little book. And I have already detected 3 mistakes. So I offer anybody who reads the book and finds a 4th mistake a dinner, an invitation to a dinner. And the last one is this sentence from Robert May, a very famous ecologist, that he said that in a congress many years ago, and I was really impressed by this. He said this: "We have a catalog of all the objects our instruments can detect in the universe. However, we ignore how many living beings share the Earth with us." And I think this is very much still true, although maybe with these sequencing techniques we are getting close to it. Thank you.
Thank you for your presentation. We have a few minutes for questions.
I was wondering, if you have an organism on the rare side of that abundance curve, what does it do there? Is it just waiting for the conditions to change? And for how long can it wait?
I think there are many different reasons to be there. As I was saying, for example, this bibliofishery, they are there waiting to find a baby squid. And of course, eventually, many of them will die. Most of them will probably die. But they're enough that the new generation of squids can get them. Some others may be forming spores or some kind of resistance stage. This has been seen, for example, for some bacteria from thermal vents in cold sediments in Svalbard. So they can, even though they can only grow at high temperatures, you can find the spores in the cold sediments, and they are there, and maybe eventually they will be coming back. And others will be growing periodically. For example, if there is a seasonal cycle, maybe they will grow in the spring, and they will remain in the biosphere for the rest of the year. Now, how long, I don't know. I suppose if you have a spore, you can be there for a long time. If you don't have it, probably not as long. But as I was saying, the main loss mechanisms for bacteria, which are predation and viral lysis, are not affecting you. And if you're not at the surface, UV radiation doesn't hit you. So there are very few reasons to die in that situation. You could be growing very slowly, for example, just maintaining your metabolism and stay there forever. And the other case is the case of the beta proteobacteria. Maybe in the ocean, they are part of the rare biosphere because they are extremely sensitive to these viruses. Maybe these viruses are very efficient at getting them, and that's another reason why they are there.
As long as they have cysts or other resting stages, one can imagine that they could survive for a very long time. But there are people who claim to have found million-years-old organisms deep in the course of sediments in the ocean. How can they survive there?
I think that there was also a calculation that some of these bacteria inside rocks, inside the crust, that they could divide once every 100 years. So these dimensions are completely different for us. There's been a lot of discussion about what is the minimal amount of energy that you need to maintain your cellular structure. Of course, this will be different. For example, if you're in a cold place, in ice, you can last a lot longer because everything is slower. If you're in a hot place, I don't know how they keep themselves, but they certainly need a certain amount of energy to replace molecules that are damaged and this kind of thing. I don't know. I don't think we know very much about this bacteria, but it's certainly fascinating.
Carlos, thank you very much for a wonderful talk. I want to go into the rear concept. Do you think rear is common everywhere? It means everything is everywhere in those terms? That is, the problem is that it is so rare that we cannot detect it. What do you think about it?
Yeah, every time you look at dispersal, it looks like it's not a problem. So it looks like even between the poles, you can have bacteria dispersing from one side to the other. If that is true, you should have everything everywhere. The question is, how rare can it be before you stop detecting it? And I think the cultures that I showed at the beginning shows that actually you can be alive and yet undetectable by current methods. Because just one more thing, having only one cell in theory is enough, and one cell among when you're sequencing 1 million sequences is nothing. So you may miss it and not find it very easily.
Yeah, but that means if you are using, for instance, the same type of protocol to culture and use the same protocol in different environments, you can get at the end the same.
You should, yeah. Yeah, I mean, if it's true that everything is everywhere, you should find them. So that's a very nice study. I'll propose it and see whether we can do it. Because, I mean, it would be a good test, because if they are everywhere, you should be able to isolate them from everywhere. Yeah.
You point out several natural processes in like transporting cells that are rare to different conditions. So I was wondering, like, maybe we are kind of pushing the system. You think like some anthropogenic effects, like for example, ballast water, can move some different type of organisms and those microorganisms. So do you think that this can have a sort of impact? Putting this together with natural processes?
I think that, I mean, ballast for invertebrates and so on, and for algae, obviously it's been a very serious problem. For a bacteria, if I have to believe the dispersal data, saying that bacteria are able to disperse anywhere, this should have happened many times through history already. Yeah, but I mean, I'm not sure about it. But if I believe that dispersal is possible, I have to accept that this has happened many times before.
In that graph that you show, where you compare the high temperature and ratings of these different I recall when you said that the ones that come from very high temperature are basically the same depending on where they come. As you move
One possibility would be that, yeah, of course, that the chemical composition of the water doesn't seem to be that important, for example. And we thought about what would be the different when you lower the temperature. And I guess what happens is that predators are more abundant as you lower the temperature. And these predators may be different in different places because they are bigger and they don't disperse as well from place to the other. So maybe this is one of the causes. But I think we need to do a lot of experiments to really show this.
With this concept of everything is everywhere, has anyone, for example, let's say one of the most abundant bacteria in the ocean is okay, there is only one person in the world who can illustrate it. Okay? Or worse.
With and without mustache?
Sorry, without the mustache. But this aside, do you think that you could take a liter of water from anywhere in the world, and if you have the skill to do it, to cultivate sour-lemon, would you think that it would be possible to get sour-lemon from any single liter anywhere in the world, including some of these experiment environments?
Yeah, I think, Linda, I think...
Or does the concept break down at some point?
It probably has its limits. I guess that's the most logical thing to think. But if I recall properly, in the ICOM database, there was one phylotype of SAR11 that was everywhere, right?
Yeah, but there weren't really well, that's
Yeah, so that was, I think that's the closest data set that we can use to answer this. But of course, if, I would assume if you move into a high thermal environment, I don't know.
And actually, there is a counter-example for cultures, for example, that they find anyone trying to isolate it from the Arctic Ocean.
They are to isolate, yeah. But the sequences can be found here and there.
In the Arctic Ocean?
Yeah.
For cultures?
I think so. No, maybe I confused with Synechococcus.
Yeah. That was, yes. So I think it's more some stuff are everywhere, but some are not.
How many profluorococcus genomes is there? And if they all profluorococcus, they're not the same profluorococcus.
Yeah, of course. Yeah.
We have now many genomes of 15 or 20.
Carlos, is this concept of where biosphere crossing all the domains, like bacteria, archaea, and microbial has this thing? Because I have maybe the wrong idea that would be more applicable to heterotrophic bacteria than, for example, to cyanobacteria. So you have Conchloropus, Cicococcus in the ocean, and I don't know if you have like a tail of rare cyanobacteria there, and I don't know if you can apply to microbial also, that you have this long tail of rare organisms.
Let's say an independent data set was a study in the Mediterranean of phytoplankton, actually of protists, because there were also heterotrophic protists and so on. And this was done by Margalev, who, besides being a theoretician, spent many hours in the microscope and could tell many different subtle differences. And so it was the only data set with enough different OTUs or taxa, whatever, and with enough individuals to compare to some of these sequences. And the shape was practically the same one. So these were eukarya. But if you take only cyanobacteria, which is just one group, I bet that if you do detailed taxonomy, you also find this kind of shape, because I think this is inevitable. But I'm not sure how long that will be. I don't know if you have any idea about that. No. But at least for the eukarya together, it does behave the same way.
More questions?
So, am I correct by saying being rare is a dynamic concept? An
Yeah.
organism can be rare because you didn't find it or didn't found in very low abundances at one time, and it can grow up and it becomes a very abundant organism. And as the conditions change, it can become a rare organism again.
Mm-hmm.
You would agree to that?
Yeah, definitely. I mean, to be there at some point in space and time, you have to grow, because eventually, otherwise you will disappear. Yeah. Yeah.
If that's the case, then you might become extinct. You would have, branches that are not here anymore, or.
Good question. Don't you think that before that happens, they will have exchanged genes with everybody else?
Right, but I think in terms of lines, genealogies, and we speak. You know that you show examples of extinctions.
Well, yeah. Well, I put the word extinction. Maybe I should eliminate that to avoid this question.
Okay. Thank you.
Thank you.